Today's topic is cultivation, growth and nutrition of bacteria. Let's get into the topic. A microbial culture is the method of multiplying microorganisms by letting them produce in predetermined culture media under controlled laboratory conditions. Microbial cultures are used to determine the type of organisms, its abundance in the sample being tested, or both. Isolation of bacteria. Properties of bacteria, that is, culturing bacteria is the initial step in studying its morphology and its identification. Maintenance of stock cultures, estimate viable counts to test for antibiotic sensitivity, to create antigens for laboratory use. Certain genetic studies and manipulation of the cells also need that bacteria to be cultured in vitro. Culturing on solid media is another convenient way of separating bacteria in mixture. Culture media. An artificial culture media must provide similar environmental and nutritional conditions that exist in the natural habitat of a bacterium. A culture media contains water, a source of carbon and energy, source of nitrogen trace elements and some growth factors. The pH of the medium must be set accordingly. Uses: enrich the number of bacteria, select for certain bacteria and suppress others, differentiate among different kinds of bacteria. Pure culture. In the laboratory, bacteria are isolated and grown in pure culture in order to study the functions of a particular species. A pure culture is a population of cells are growing in the absence of other species or types. A pure culture may originate from a single cell or single organism, in which case the cells are genetic clones of one another. Pure cultures are obtained by using variety of special techniques. All glassware media and instruments must be sterilized, that is, aseptic techniques are used for obtaining pure cultures. Basic requirements for obtaining a pure culture are solid medium, a media container that can be maintained in an aseptic condition, and a method to separate individual cells. A single bacterium supplied with right nutrients will multiply on the solid medium in a limited area to form a colony, which is a mass of cells all descended from the original one. Agar. Agar, a polysaccharide extracted from marine algae, is used to solidify a specific nutrient solution. Unlike other gelling agents, it is not easily degraded by many bacteria. It is not easily destroyed at higher temperatures, and therefore it can be sterilized by heating the process, which also liquefies it. One solidified agar medium will remain solid until the culture media is contained in a petri dish, a two-part glass or plastic covered container. Classifications of culture media. Bacterial culture media can be classified in at least three ways: consistency, nutritional component, functional use. Classification based on consistency: liquid media, solid media, semi-solid media. Classification based on consistency: liquid media. These are available for use in test tubes, bottles or flasks. Liquid media are sometimes referred as broths. In liquid medium, bacteria grows uniformly, producing general turbidity. No agar is added. Mostly used for inoculums preparation. Solid media. An agar plate is a petri dish that contains a growth medium used to culture microorganisms. 2% of agar is added. Agar is the most commonly used solidifying agent. Colony morphology, pigmentation, hemolysis can be appreciated. Examples include nutrient agar and blood agar. Semi-solid media. Such media are fairly soft and are useful in demonstrating bacterial motility and separating motile from non-motile strains. Examples of semi-solid media: Hugs and Liefens oxidation fermentation. 0.5% agar is added. Classification based on nutritional components. Simple media, complex media, synthetic or chemically defined media. Simple media such as peptone water, nutrition agar can support most non-fastidious bacteria. It is also called as basal media. Example: NBNA. Nutrient broth consists of peptone, yeast extract and NaCl. When 2% agar is added to nutrient broth, it forms nutrient agar. Complex media. Media other than basal media are called complex media. They have special ingredients in them for the growth of microorganisms. These special ingredients like yeast extracts or seasoned hydrolysate, which consist of mixture of many chemicals in an unknown proportion. Synthetic media or chemically defined media. Specially prepared media for research purposes where the composition of every component is well known. It is prepared from pure chemical substances. Example: peptone water. Classification based on functional use or application. Enriched media, selective media, differential media, transport media, indicator media, anaerobic media. Enriched media. Addition of extra nutrients in the form of blood serum, egg yolk, etc. to basal media makes them enriched media. Media used to isolate pathogens from a mixed culture, stimulate growth of desired bacteria and inhibit growth of unwanted bacteria. Media is incorporated with inhibitory substances to suppress the unwanted organisms, thus increase in numbers of desired bacteria. Examples of enriched media: chocolate agar, blood agar. Selenite F broth for the isolation of Salmonella shigella. Tetrathionate broth inhibit coliforms. Alkaline peptone water for Vibrio cholerae. Chocolate agar. Chocolate agar is a non-selective enriched growth medium used for growing fastidious bacteria such as Haemophilus influenza. Blood agar plate contains mammalian blood, typically at a concentration of 5 to 10%. PAP or enriched differential media used to isolate fastidious organisms and detect hemolytic activity. Selective media. The inhibitory substance is added to a solid media, thus causing an increase in number of colonies of desired bacteria. Selective media and enrichment medias are designed to inhibit unwanted commensals or contaminating bacteria and help to recover pathogens from a mixture of bacteria. Any agar medium can be made selective by the addition of certain inhibitory agents that don't affect the pathogen. To make a medium selective include addition of antibiotics, dyes, chemicals, alteration of pH or a combination of these. Examples of selective media: Thia-Martin media, selective for Neisseria gonorrhea. EMB agar, Camelophytic agar. Differential media. Certain media are designed in such a way that different bacteria can be reorganized on the basis of their colony color. Various approaches include incorporation of dyes, metabolic substrates, etc., so that those bacteria that utilizes them appear as differentially colored colonies. Substances incorporated in it enable it to distinguish between bacteria. Transport media. Clinical specimens must be transported to the laboratory immediately after collection to prevent overgrowth of contaminating organisms or commensals. Delicate organisms may not survive the time taken for transporting the specimen without a transport media. This can be achieved by using transport media. Examples of transport media: Carey-Blair medium for Camelobacter species, alkaline peptone water, Stroud's medium, buffered glycerol. Anaerobic media. Anaerobic media need special media for growth because they need low oxygen content, reduced oxidation, reduction potential, and extra nutrients. Thank you.